Hello class, today I'll be talking to you about the effects of phenotypic variation in blackberry flowers on pollinator activity, here from the field. Have you ever wondered why bees like flowers? And if so, why not take it further? What specific features of flowers do bees like? Well, you're in luck, as I'm about to tell you. But blackberry flowers, as seen growing on these trees right behind me, are an invasive species here in Australia, and are actively spread by the western honeybee. But what is it about these blackberry flowers that bees like so much? This yields a perfect opportunity to explore the effects of phenotypic engineering on the European blackberry flower, and the resulting effects on pollinator attraction and loitering times. But what is phenotypic engineering? Phenotypic engineering is the changing of certain physical elements of an organism to express different features than it naturally would. This can come in the form of changing their shape, their size, or their color. But but why do we need to know this? You're asking yourself. Why do we need to study these flowers and these bees? Well, to put it simply, there is a knowledge gap. There is a clear knowledge gap between the relationship of the European blackberry flower and the western honeybee. There has been ample research conducted on how differing floral traits affect pollinator activity in other flora, as well as research on the effects of altering phenotypic features on pollinator attraction as well as loitering time. Additionally, research has been conducted about which phenotypic features bees favor and their role as pollinators. However, this study will address the knowledge gap by investigating the effect of phenotypic variance of blackberry flowers on attraction as well as loitering time together. Understanding this can lead to a framework that can be applied to other invasive species, allowing development of effective control and prevention methods, as well as it can be applied in agricultural settings to increase yield of blackberry plants. To answer this research question, 5 different blackberry flowers were altered to express 5 different phenotypes. These included a normal flower, a flower with 2 reduced petals, a flower with 2 additional petals, a round flower, and a yellow flower. These were all accomplished in different ways. The flower with 2 less petals was achieved by using sharp scissors to remove petals. A flower with 2 extra petals was achieved by using a non-toxic, non-odorless glue to attach flowers' petals from a surrogate flower. The round flower was attached by using a thin sheet of cellulose, which was cut in a circle and attached to the back of the flower, as well as the yellow flower, which was achieved by using a turmeric concentrate diluted in water applied to the flower through a pipette. This research question was explored using an experimental unit. This experimental unit consisted of 5 flowers in a 60 by 60 centimeter area with no other flowers present, which each of the flowers altered to match one of the corresponding phenotypes. This experimental unit was monitored for 30 minutes with 5 different stopwatches, each corresponding to a different flower. This was used to measure the bee loitering time. Once a bee entered 1 centimeter of the flower, it was deemed as loitering and the stopwatch was started. What was also recorded was the bee attraction, so how many bees visited the flower as a whole. 5 30-minute observational periods were conducted over 5 different days, and at each one of these days the weather was recorded, which included the wind direction, humidity, temperature, as well as the general weather conditions. The results indicate that there is a very little difference between bee loitering times as well as bee attraction rates between the different phenotypes tested. However, yellow flowers and flowers with -2 petals have the longest average loitering time of 153 seconds compared to that of the control averaging only 145. For bee attraction, the flower that had -2 petals featured the highest average with 22 sightings, and yellow with 17 sightings, and the control only averaging 13. As seen here with the 5 different treatments modeled against the time, you can see that yellow petals as well as the -2 petal flower had the highest rates of average loitering time compared to the others. As well as in this graph, you can see the 5 different phenotypes modeled against the number of bee sightings. As you can see, -2 petals and the yellow flower had the highest average amount of sightings compared to the other flowers. There is a significant amount of knowledge gained by conducting this experiment, as it can act as a framework which can be applied to other flora and fauna. As there has been no previous research conducted around blackberry flowers specifically, especially with phenotypic engineering, as well as the effects of blackberry flowers on pollinator activity. There has been very little research conducted surrounding blackberry flower and how pollinators play such a vital role in spreading this invasive plant. The study can benefit the community by identifying an ideal phenotype that flowers can be altered to increase pollinator activity to increase yield in an agricultural setting. It also can identify an unappealing phenotype that the species can be altered to reduce pollinator activity to reduce the spread. Reducing the spread and controlling the blackberry flower is essential, as the economic impact of noxious weeds in Australia is estimated at $3.9 billion, with the government spending $116 million a year on noxious weed control, saving you money on your taxes.